What is up, everybody? Welcome back to another episode of Wildlife Cake and Cocktails. We have a fantastic show for you today. We're going to be speaking with Johanne Martens from the Deakin University down there in Victoria at the Centre for Integrative Ecology. And in honour of her, our topic for today, largely on birds, I have something called a black feather. Wonderful. So that is cognac, triple sec, and a lemon twist in there, along with a bit of vermouth. So it's a fairly dry drink. Oh, also got some lovely Angostura bitters in there. Fairly dry and bitter drink, but very, very delicious. And a lovely slice of hazelnut cake, hazelnut chocolate cake here in front of me as well. All because we're going to be talking about feathery things, birds, specifically parrot diseases, more specifically Beak and Feather Disease here in Australia, which is the subject of the PhD of our main guest for today, Johanne Martens. She is down at the Centre for Integrative Ecology at the Deakin University in Geelong, doing her PhD since 2016 onwards. Her research interests include pathogen transmission dynamics, breeding biology and behavior, and conservation in birds. Following a Bachelor of Science at the University of Hohenheim in Germany, she completed her Master's in 2014 between the University of Hohenheim and the Max Planck Institute for Ornithology. This was followed as a position in research as a research associate at the Max Planck Institute of Ornithology, also spending some time at the State Museum of Natural History in Stuttgart. She's obviously now, since 2016, down at Geelong at the Deakin University, studying ecology and evolution of Beak and Feather Disease virus. That's a BFDV for short, in Australian parrots. You can follow her at Johanna_Martens on Twitter, and you can also check out the Deakin Centre for Integrative Ecology at Deakin CIE. Let's bring her in now. Hi, Johanna. How you doing?
Hi, I'm good, thanks. How are you?
Yeah, very well. A little bit cold here this lovely wintry day, but I guess a little bit colder down south where you guys are at in Geelong.
Absolutely. It's not as bad today, but last week we had a lot of frozen windscreens and these things.
Yeah, right. So I grew up in the Dandenong Ranges, and I do remember, it can be pretty chilly. So I guess getting straight to the point for you guys down there in Victoria, I guess spending a little bit more time inside than usual with everything going on with COVID. How are you guys managing? I understand that you're not in the center of the lockdown area, but not too far away.
That's true. It's a bit of a worry that we are back into lockdown in some areas, but we are quite lucky down here in Geelong because we are not in lockdown currently, so there's still lots of time for exercise outdoors, which for me is mainly bird watching. So that's
right.
Good.
Right. And obviously I imagine bird watching, you kind of get away from some of the more populated areas where there's a bit less chance of disease transmission from other people.
Exactly. It's the best hobby to have for social distancing.
Yeah, fantastic. Yeah, well, yeah, same thing with herpetology, going out looking for reptiles. You kind of, not exactly doing it in big groups in enclosed spaces, but I guess you must have a bit of an understanding as well of pathogen and disease transmission, having such an interest in viruses and disease ecology. Has that been something that you've been interested in for a long time, or is this a bit more of a recent thing for you?
Well, I come a bit more from the bird side of things with my research, but for the last 4.5 years almost, I've been working on a virus in birds. So that, of course, included a lot of reading about viruses and pathogens in general. So I now know a bit more about it. But before that, I was mainly focused on other aspects in birds.
Right. So I can see from some of your researches, you really had like an ornithological, focus, particularly early, with some very fascinating research in the early 2010s in Stuttgart, in Germany, on the invasive Amazon parrots, the yellow-headed Amazon parrot, Amazona oratrix, and the blue-fronted Amazon parrot, Amazona aestiva xanthotera. ducks, if I get that right. And they also hybridize in their invasive populations in Germany. Is that correct?
It's correct that they hybridize. It's not correct that they're invasive, actually. So invasiveness has to kind of be monitored and checked for each new introduced species. And with the work I've been doing with the Amazons, we couldn't actually see that they were invasive, so they were not displacing or being a threat to the native species.
Right.
They seem to have just found their own little niche, and just in the city of Stuttgart, which they haven't left in over 30 years, and don't seem to be much of a threat. So we call them introduced.
Right, introduced versus invasive. So invasive having much more of a negative impact and competing with locals.
Exactly.
That's fascinating. That something from the Amazon would have a thermal niche in somewhere as cold as Stuttgart. Did you guys find that surprising, or is there some kind of behavioral or maybe microclimate preferences that they exhibit in these introduced ranges?
That was the very fact that really fascinated me about this flock, because the very first time I got to see them, someone showed them to me, was when it was getting dark in winter at their roost site. It was snowing heavily, the branches were covered with snow, and suddenly these parrots come in, bright green and red and yellow, and scream around and just bring a bit of tropical feeling into the winter, and just fall asleep in the snow. And they survived that, and they have been surviving that for a long time. So that's actually what fascinated me from the start, and that's why I really stuck with them for a long time. So I started working in them for my bachelor's thesis, which is usually just 3 months, and I extended that to 9 months, and then I kept going for another 4 or 5 years, just because there was nothing known about how they survive in this super novel climate and environment. And there are just lots of different little factors that make Stuttgart bearable for them.
Yeah, interesting. I mean, the juxtaposition of that tropical vibe with snow would be a little bit mind blowing. It's kind of like going into a tiki bar when it's snowing outside, or like getting some tiki drinks at the ski lodge.
Pretty much, yeah. That's a good description of it.
Yeah, fascinating. And you guys did publish several papers, some one on diet and feeding behavior, and one on nest site selection and reproduction. Did you guys find any? For me, I guess the nest site selection would be fascinating. Did you guys find any difference in how they select their nest sites in their introduced range?
It was quite hard to compare because there's actually not much data available from the native range. Especially yellow-headed Amazons are endangered, very rare in the wild. The nests very hard to access in these dry forests in Mexico. So for us, it was a lot easier, although it was still a lot of work to find the nests, to access the nests, and to collect data. But it looked like the nests were a bit lower down in the trees, for example. So we found some differences, but it also has to do with availability, because they're pretty large parrots. They're about the size of a crow, and they need really big tree hollows to nest in. And they just have to make do whatever's there as well. So that limits their choice a bit.
Interesting. Well, I guess maybe if they're selecting something lower, it might be a bit less exposed to some of the winds and blizzards. And being a big bird that needs a big hollow as well probably has a bit of thermal buffering, I imagine.
May do, yes. And what helps is we couldn't actually measure that, but what's known from other parrot species or hollow nesting birds is that the tree itself can insulate the nest pretty well, especially if it's a hollow in the tree trunk. The temperature often is not that different between the seasons.
Yeah, absolutely. We definitely know here from Australia that a lot of animals do very, very well in termite hollowed or hollow bearing trees in general. It seems to be one of the major niches of a lot of our arboreal species. So yeah, kind of unsurprising, but still very, very fascinating. Sorry, go on.
No, it was really fascinating because it's so different for them. They just live in urban parks and they frequent the zoo as well. And I guess Stuttgart's pretty special because the city has these big, very old parks, with ancient, huge trees, mainly London planes. which are the most important trees for nesting for these parrots. And they just don't find that outside the city. And I think that's one of the main reasons why they don't really spread into the surrounding countryside, because they just need these huge trees with huge hollows, like a lot of other big parrots around the world. And like everywhere around the world, these trees are declining. And also what is another special feature of parks is that there are a lot of exotic planted tree species that the parrots can feed on year round, which they also just don't find in the countryside.
Yeah, fascinating. I guess just in terms of impacts, you mentioned that there's not really a lot of competitive impacts or direct, invasive sort of typical impacts as we understand them, competition, direct predation for a lot of introduced predators and things like that. We should probably get to our main topic. Do you suspect that there might be any impacts of disease vectoring, having maybe slightly concentrated clusters of these birds in urbanized areas? We know from some of the work done with house finch disease, avipox virus, particularly in the book by Professor Darryl Jones from 2019, Feeding the Birds at My Table. High density activity at feeders and things like that can be a way that certain diseases can spread. I guess something like that would be pretty hard to monitor in these introduced populations, but do you suspect that there could be the potential for something like that to occur from these invaded populations to certain native species in Germany? And I guess secondary question, is there any closely related parrot species in the area that would be a likely host for something to be transmitted to?
I think it could definitely be a problem that urban species or species in general nowadays are closely together in high densities in small remaining habitat fragments. I think that's a problem in general. I'm not sure how much of a problem these introduced birds would be. They often escape pets that are probably, they have been vaccinated and medicated before they maybe escape. There may still be some disease jumping around. I would love to look at that. I'd love to compare urban and rural parrots for that, and haven't had the chance to do that yet. But in Germany, we don't have any native parrots, so there are no close relatives there. We have introduced ringneck parakeets in much larger numbers all throughout Germany, and there has been some disease found in them, but you'll also find probably a lot of different pathogens in all sorts of other wild birds. Just think of avian influenza, for example, in all the water birds. It's just quite common. And the more closely together everyone gets at feeders in urban areas, people feeding parrots, all these things, it doesn't really help with social distancing. And then it just increases transmission rates.
Yeah, absolutely. And I guess it's something that we also need to remember is that diseases, viruses, pathogens, bacterial, fungal, whatever they are a natural part of the population and are always going to be there at some kind of background level. It's really these massive spread events where things go and kind of break out of small populations and become spread really far and wide that we start seeing those bigger impacts, as has been shown with house finch disease, which is it's a mycoplasmal conjunctivitis, I believe. So is that a protozoan disease? There's
the
bacterial, there you go. And then there's the avipox virus that we spoke about, which is a pox virus, and of course there's the beak and feather disease, a cytosine circoviral disease. So, there's quite a variety of diseases out there, some of which kind of fluctuate from, I guess, background levels to slightly higher. And I guess that impact of disease vectoring around concentrations is always going to be a concern.
Absolutely, yeah. Because pathogens, they evolve, they mutate, they can jump to new hosts, and living in close vicinity with other animals and so on and so forth just facilitates these host jumps. And it's a bit like if you still go to work when you have a cold, your colleagues will get it, whereas if you stay at home, you won't. Same as with COVID now.
Yeah, and I guess birds as well. Obviously, harder to lock them down to one area because they can move so far and so easily by flight. A little bit harder to maintain, vectors and pathogens from being transmitted from one area to another when they can just fly away. And I understand that a lot of these diseases can be spread from even just respiratory droplets, but that's for things like housefinch disease and whatnot. What about beak and feather disease? Is it more of a feather and dander kind of transmission, or do we know if it's spread respiratorily as well?
It can be spread through a lot of different pathways, really, which is part of the problem. So contaminated feather dander, feather dust, like you just mentioned, but also through crop secretions. So when birds feed each other, parents their young, or partners feed each other, or maybe even saliva at feeders, although we haven't investigated that yet. The virus is very stable outside the parrots in the environment, so it is fairly likely that in some cases it can be transmitted through contaminated environments, like feeders, for example. It can even be transmitted from mother into the egg. So feces, so it's quite easy to spread it.
Right. So yeah, transmits fairly easily. I guess we should give our listeners here a bit of a background on Beak and Feather Disease virus, just in general. So this is from Martens et al. 2019, one of your papers in EMU, the Austral Ornithology Journal. Persistence of beak and feather disease virus infection in wild crimson rosellas, Platycercus elegans. So just taking a little bit of background on the beak and feather disease virus. It's a single-stranded DNA circovirus, pretty much with a global distribution. Many species impacted from one of the most threatened bird orders, the psittaciforms, which is the parrots, cockatoos, and lorikeets. Did I get that right?
Yes.
Psittaciforms. Recently found in non-psittacine birds as well. And horizontal transmission through feces, feather, dander, crop secretions has been noted, and vertically from mother to egg. Mostly fatal in young birds, chronic and often fatal in older birds. However, there's limited data about persistence of beak and feather disease in wild populations. This was actually a great paper. You guys studied over 14 years, 55 wild crimson rosellas in Victoria, testing blood and feather 2 to 5 times for beak and feather disease and the antibodies, and looking at infection persistence. And if birds were initially positive, they were likely to show positive about 5 months after capture, but declined to about 8.3% in samples after 5 months. So interesting. It shows a decline and an immunological response upon recapture. And I guess that rosellas can clear this virus, which is a great result, but I guess should test some of the broader implications and other species in which that can occur, and rates of reinfection. Fascinating paper. I guess, did you guys look at some of the non-lethal impacts as well on things like fitness and reproduction?
Yes, we did. And so this study you've just mentioned, I was actually really lucky to be able to use these data during my PhD, because I've been working on the virus for 4 years, but my research group has been doing crimson rosella work for much longer. So we had this great data set of hundreds and hundreds of birds, and then I could pull out the subsets of birds which had gone into the trap more than once, which is usually really, really hard to do. So most studies are really just with catching a bird once, testing it, and then you never see it again. So it really helped to see what happened in this subset of birds. And it's really hard to look at the effects. We were happy that for the first time we could show that wild birds can mount an antibody response and clear the virus, at least from the blood. We don't know if it can still hide out in other organs, because for that we obviously have to kill the birds to check the organs. What was the second part of the question?
Looking at non-lethal impacts in fitness and reproduction in crimson rosellas. Did you guys get much of a chance to look at that?
Yeah, right. So we did look at these non-lethal impacts by measuring body condition and also some blood. We looked at body condition, and we also did some blood tests to look at fitness effects, and we also looked at the reproduction. We didn't really find much of an effect on either. And with the reproduction, it had to do with the really, really low prevalence we found in breeding birds and their nestlings, which is great for the birds, but it meant that we just didn't have the statistical power to compare the reproductive output of healthy and infected parental birds.
Yeah, I guess that's always one of the challenges with study design, right? Like even if you, it's already hard to come into a long-term study, right? Like who comes into a study and has a 10-year data set ready to go. That's really, really fortunate. And even then it's hard to have enough samples to reach statistical significance. The joys of wildlife science.
Absolutely. Two field seasons, 2 years in the field, 2 breeding seasons, hundreds and hundreds of samples from breeding birds and the nestlings alone. It was probably over 700 samples, and then I think 11 turned up positive, and half of them were parents and half of them were nestlings. So I have 5 nests with an infected parent. There's not much I can do with that statistically, but that's just what it is in the wild. That's just what we get.
Yeah, and that is still the result. It is still a great result from the monitoring, and to show that that this bird can clear this disease and deal with it fairly well. It seems to be prevalent and fairly present in fairly low amounts in the population. So it's not like it's going pandemic on them. They don't have their own massive issue with it. But I guess one of the other issues that came out is can latent beak and feather virus in Rosellas, for example, can it spread to other populations that might be at risk, right? Such as swift and orange-bellied parrots that they share habitat with, which, for those threatened species would obviously be much more of a concern than it would be for something quite as common as crimson rosellas. It's still concerning, but yeah, in those threatened species would be much more of an issue.
Exactly. For example, this year we did another study where we screened a range of abundant parrots, so not just crimson rosellas, although they were included as well, but also galahs, silver-crested cockatoos, and then also blue-winged parrots as a bit of a special case because they're really closely related to orange-bellied parrots and occur in a similar habitat. And it just wasn't known whether they have BFDV and how it affects them. And we found, so in total we tested 7 species, although we focused on the 4 I just mentioned because we caught those the most often. And we found BFDV, so Beak and Feather Disease virus, in 6 of those 7 species. And crimson rosellas had the highest prevalence again. And when we had a closer look in a different study at crimson rosellas, we could see that especially in sub-adult birds, young birds that are less than 1 year old, the prevalence can actually be really high. It can reach over 60-70%. But we never see signs of disease. So in all of these birds that I just mentioned that we screened, we didn't find sick birds. We had 1 or 2 sick birds, but I actually excluded them from the study because I just wanted to have healthy looking birds that were all trapped with the same method, because once they show signs of disease, I can sometimes catch them by hand because they can't fly anymore, which kind of biases the data.
Yeah, right.
And so, but the take-home message there is that we had a high prevalence and a wide spread of the virus in absence of these clinical signs, which I found really fascinating.
Yeah, right. So it very frequently is, I guess, not non-symptomatic, almost like the pandemic that we're dealing with COVID-19, right? That probably some of the greater amounts of transmission happen from people who are asymptomatic. I heard there was potentially 1 case of an asymptomatic person passing the disease to maybe over 50, 60 people in one instance. Yeah, so I guess we hope it's not quite as virulent as COVID-19, but the fact that you mentioned that you can catch a lot of these animals by hand once they're infected and really suffering the impacts. It seems to be quite a significant disease. Have you seen a lot of the clinical impacts of it on these birds during your time? And what does it look like? And I know it's tough working with injured and sick wildlife. How has it been for you seeing the impacts of a disease like this on an animal that you really enjoy?
Well, luckily enough, I didn't often find diseased birds. You sometimes see them around, even if you just go on a trip somewhere where cockatoos get fed by humans, and you just get up closely to them, and you sometimes see a sick one. And that is always a huge pity. Even when people contact me, and I sometimes get messages where people ask me, what do I do with this bird? Is this Beak and Feather Disease? And there's just nothing we can do, and it's just pretty sad and awful, but it's kind of a driver for my work. I want to work in conservation. I really, I really like doing something where I could maybe have a positive impact or help the birds a bit. The disease, when it breaks out, or so when there are signs of infection, it can be pretty severe. I've mainly here in Victoria seen it in sulfur-crested cockatoos, and they start by looking really grubby, so they are not this beautiful pure white color anymore, but they start looking dirty and scruffy. And then with the chronic form of the disease, they lose more and more of their feathers. They can't, during molt, when they usually grow new feathers, that feather growth just doesn't work properly anymore, so the feathers fall out. And then the claws and the beak also don't grow properly anymore, so they get quite severe beak deformities sometimes. So they start looking really, really rough. So they are more exposed to the elements because they lack their feathers. They can't eat properly anymore because of the deformed beak. And they also have a lot of internal damage, so their organs and tissues can get affected severely. So the immune system is often affected, so they often die due to secondary diseases because the immune system doesn't work well anymore, so they are more susceptible to other diseases which they then die from. So it can get pretty awful when the bird actually gets the disease.
Right.
In crimson rosellas, I've never actually seen severe signs of disease. They may be quite resistant or tolerant to it because they have these high prevalences, but fairly low intensity of infection, so viral load, which we can measure as well, amount of virus in the blood. And they don't show these severe signs, whereas in cockatoos, once they get it, even when they don't show signs yet, they have massive viral loads, and then they get these massive signs. So it's just probably different ways of dealing with the virus. And it's a bit similar to COVID almost, where you can also see like that very old people, for example, older people are more affected by it. So you really have a very specific impact, and we see that in the birds sometimes as well, that the very young birds are most affected because the immune system is not as well trained yet, as experienced, if you want to put it that way. And so it's really important to look really closely because you have all these species and age and sex specific impacts or breeding status effects that, when we suddenly split up our birds in breeding and non-breeding subsets, we had very, very different prevalences again. So there are so many factors influencing it that it gets quite complex.
Yeah, I can imagine. But again, interesting to hear some of those, I guess, similar things to COVID. Like it affects people with obviously weaker immune systems, like the elderly or older birds, or younger birds in this case with a non-developed immune system, which is a bit different from COVID. But again, the comorbidity with other viruses, lowering your resistance to things like pneumonia and then having secondary complications from these kind of diseases is very, very common with a lot of I guess respiratory viruses. So and I guess all of this is really important in terms of controlling some of these risks for bird epidemics for wildlife carers, right? So I suppose a lot of wildlife carers are going to be kind of on the front lines and likely to maybe see some of the visible impacts in Australian wild birds. So knowing what some of those impacts look like, the dirtier coloration, the loss of good feathering, the beak and claws, those visible impacts I guess are really, really important to know. If wildlife carers out there do see these kind of impacts, Is there a response that you recommend? Obviously, with a lot of viruses and a lot of diseases, you don't want to have too much contact, and you don't really want to spread it around too much. So maybe grabbing the bird and bringing it into care isn't necessarily the number one move, but reporting it and monitoring and stuff might really be very important for controlling this disease. No?
I think that'll be really difficult. Especially wildlife carers will come into contact with it, definitely. And most of them are probably aware about the very good hygiene they need to practice, and that bird needs to be quarantined. Most of the time they unfortunately get euthanized because there is no treatment available usually, and once that bird is so sick that you see severe signs, it's probably suffering a lot. And but it seems to be really widespread here in Australia, and often without any signs, so there's not much we can do in terms of reporting and controlling, I guess. But it is super important, whether you care for birds or feed birds at a bird feeder in your garden, or you have a water bath, just to practice really good hygiene and clean everything really, really well to just prevent the spread as good as possible.
Yeah, right. So minimizing the risks of transmission is probably the more important thing.
Exactly, yeah, because even the larger populations can be affected by it, and it becomes a particular problem if it spills over into threatened species. And in these, when sick birds get brought together in wildlife shelters or other, in close contact in captivity, the virus again can mutate. You can create new virus strains, then you release the birds again, you bring this new strain into the wild, and that can be a higher risk again, because the birds out in the wild don't know this strain, the immune system doesn't know it. So yeah, preventing the spread and the spillover, especially to threatened species, is super, super important.
Right. And we're back to social distancing.
Yeah, exactly.
Yeah. So, as mentioned in Professor Darryl Jones's book, some of those risks do, I guess, increase in urban environments where people have, fairly dense feeding practices. So, backyard feeding practices and cleaning are really, really important. Using, lesser amounts of food, giving less frequently and maybe more irregularly, so you don't get big communal flocking at the same time of large amounts of birds. And cleaning, using a little bit of vinegar and water and a cleanable, removable, replaceable feeding surface rather than just the same old dish covered in bird poo that you're putting bird seed into, and things of that nature, I suppose.
Exactly right. Yes, because as we've just seen with COVID, we also just with any virus, we don't know what it'll do. So that's why we're trying to learn as much about them as possible before outbreaks happen. But it's always better to be careful when being in touch with wildlife. While it is, of course, important and fascinating to be in touch with wildlife, but good hygiene is definitely important. And I mean, there are other pathogens out there too that are already zoonotic, that already can jump to humans. And if Beak and Feather then, facilitates secondary infections with maybe these zoonotic pathogens, there's always a bit of a risk that you can get infected with something. So being really clean about it is definitely a good approach.
Yeah, absolutely. And I guess keeping an eye on future risks and all the monitoring and science that you guys do might help keep an eye on diseases such as this. But do you have maybe any suspicions of what might happen with future risks, such as modified distribution or increasing habitat loss and maybe even things like climate change? Do you suspect that there's going to be any specific impact of these on Beak and Feather Disease or other diseases in the future? I know that's a bit of an open-ended question, but
yes. Well, habitat loss and climate change will definitely also affect pathogens. Habitat loss means that animals get squashed together into smaller habitat fragments. They're already stressed, they're already weakened, maybe because they don't find enough food, maybe there are not enough nest hollows, maybe they fight more about remaining nest hollows, which again increases contact between the birds. We already know for other pathogens, for example, malaria, that in some areas of the world, We don't have malaria yet because the vector mosquito can't survive in these areas yet because it's too cold. Tourists bring it back, but it doesn't spread because the mosquito isn't there. But if it gets any warmer, and the mosquito will be able to survive. And that gets brought in accidentally as well with the luggage or with other cargo. So then we may get malaria in areas that are now too cold for it. And those are just a few examples. So these shifts in temperature and humidity, and then together with the habitat loss, which stresses animals and other organisms, we'll definitely see an impact of that. And I mean, COVID is already a bit of a result of that. You know, animals and humans being very, very close together. For example, in these wet markets, there's not enough distance there and not enough hygiene. Everything's crowded. Things spread easily.
Yeah, and again, potentially having, I mean, there's many suspects for the kind of direct intermediate host between us and bats, but again, another very mobile, flying, and also communally gathering organism in bats, which is the potential original host for COVID-19.
Yes, and there are many viruses that are related to COVID-19, other coronaviruses. There's a whole range of coronaviruses in birds, in other mammals. This is not a single little mutant. There's just a whole range of these organisms around, and
I mean,
these things will happen again.
Yeah, including even things like the current Delta coronavirus is going on in pork populations for human consumption, which are very, very common. All stuff that we, I guess, need to keep an eye on, and with more risk is all likely to be a little bit compounding. But we're happy that we at least have a few people like yourself doing all the science and monitoring to keep us ahead of things on things like Beak and Feather. So hats off to you guys. Let's what do you think? Shall we move on to a couple of bits of new research here, starting with one of your own?
Sure.
All right, no worries. We're going to go straight to Beak and Feather Disease, and I'm going to bring this up for you guys. Bear with me. All right, here we go. And we can all see this. Very good. This is in PLOS1, Beak and Feather Disease virus prevalence, load, and excretion in 7 species of wild-caught common Australian parrots. I believe this is the one you spoke about earlier, correct?
Yes, that's right.
Wonderful. All right, I'm just going to go through the abstract here very quickly. Pathogens pose a major risk to wild host populations, especially in the face of ongoing biodiversity declines. Beak and Feather Disease virus can affect most, if not all, members of the largest and most threatened bird orders in the world, the psittaciforms. Signs of the disease can be severe and mortality rates high. Its broad host range makes it a risk to threatened species in particular because infection can occur via spillover from abundant hosts. Despite these risks, surveillance in locally abundant wild host species has been lacking. We used quantitative PCR and hemagglutination assays to investigate BFDV prevalence, load, and shedding in 7 abundant host species in the wild in Southeast Australia: crimson rosellas, eastern rosellas, galahs, sulphur-crested cockatoos, blue-winged parrots, rainbow lorikeets, and red-rumped parrots. We found BFDV infection in clinically normal birds in 6 of the 7 species sampled. We focused our analyses on the 4 most commonly caught species, namely crimson rosellas, sulphur-crested cockatoos, blue-winged parrots, and galahs. Species but not sex was a significant predictor of prevalence and load. 56.1% of positive individuals were excreting antigen into their feathers, indicating an active viral replication with shedding. Being positive in blood samples predicted shedding in crimson rosellas. Our study confirms that BFDV is endemic in our study region and can inform targeted disease management by providing comparative data on interspecies variation in virus prevalence, load, and shedding. Awesome, fantastic paper. Kind of looking at this wide number of species with potential to jump over. There we go. There's our targets right there. So tell us a little bit about this paper. Obviously, crimson rosella is fairly easy to find in Victoria, but the others might sulphur cresteds as well. But the others might have presented a bit of a challenge to survey.
Yeah, that's right. Crimson rosellas are our main study species and have been for many years in my research group. we then started, my colleague and I, to start catching these other species, and we had to kind of optimize the traps first, so that was a bit of an adventure. And sulfur-crested cockatoos and galahs are also really common around here, as are rainbow lorikeets and eastern rosellas and the red-rumped parrots. But we again had to try out what comes into our traps, what can we deal with in the time we have. And every species is so different that we then focused on those first 4 species that we could catch within a reasonable time frame and good numbers. And then the blue-winged parrot was a bit of a special case and a bit our biggest challenge, because there's just not much known about that bird really at all. They're around, they're fairly abundant, they're partly migratory, they're really evasive, they're incredibly hard to catch. So it was good fun trying, and at least we got 17 of them, and we found BFDV in them. So that was really interesting for us, especially because they're fairly similar and in the same genus as our critically endangered orange-bellied parrot.
Right. So that must have been, a great result. Obviously, you can see it there right in the middle of our screen. We got 17 blue-winged parrots sampled. And let's move on down here and kind of see the host species. And there we can see our Rosellas with a fairly high amount there, and same with the galahs. So they ended up having some of the highest prevalence in the galahs and the Rosellas. Is that correct?
The silver-crested cockatoos and the rosellas. Yeah.
Oh, sorry. Yeah.
Yeah.
Wonderful. And any significant results out of this one that you found most interesting?
Yeah, pretty much what I mentioned earlier, that it's just so widespread in absence of clinical signs. So many birds have it. They have different ways probably of dealing with it. For me, it was great that I could use different types of samples for each bird. So we got a bit of a detailed picture, because I think a lot of the virus we would have overlooked if we just screened blood samples, for example. But because we screened cloacal swabs as well, and we then did this other test, this hemagglutination test, to detect antigen in feathers, we could also really see if the birds excrete antigen, because testing the blood with the PCR assay or the swabs with the PCR assay only tells me that there's virus DNA present, but there's a small chance that some of that could be just remnant fragments of viral DNA, although it's been shown in other studies that most of it is actually infection. But that other assay in the feathers definitely will only pick up viable infectious virus particles. So then we can see, okay, they actually shed infectious particles in their feathers. And only a bit more than half of them did that, whereas the other almost half of the birds were positive in their blood samples, but they didn't shed the virus, which may be that they only got infected recently, or that they're already fighting the virus, or the virus is currently not replicating. It's a so-called latent stage where it's a bit dormant. But yeah, as you see, there are just lots more open questions. So it's just open up so many more questions, as always. But that keeps it interesting, I guess.
Yeah, no, fascinating. And so that hemagglutination study was really what showed the active transmission from the feather dander. Very, very cool. Let us move on for our next paper here. We shall go back to this and jump over to Morrison, 2019, low innate immune gene diversity in the critically endangered orange-bellied parrot. So Neophema chrysogaster, so the relative of the blue-winged parrot that we were just talking about before. Straight through the abstract here, we have outbreaks of disease in threatened species can increase the likelihood of extinction by reducing population size. Low immunogenetic diversity can increase susceptibility to and severity of disease outbreaks. Our study measured genetic diversity at innate immune genes, the toll-like receptors in a critically endangered Australian bird, the orange-bellied parrot. Conservation of this species has recently been challenged by several disease outbreaks. We found low diversity at 6 TLR genes in both captive and wild orange-bellied parrot populations, similar to results seen in other threatened bird species. 3 loci were found to be morphometric across all samples, including birds which were recorded to have been involved in recent disease events. Phylogenetic analyses of TLR sequences from Neophema chrysogaster, all as well as representatives of all other species within the parrot genre Neophema and Neocephodus, revealed a small number of sites under pervasive or episodic positive selection and a number of sites under negative selection. This study is the first to investigate functional genetic diversity in the orange-bellied parrot. Fascinating stuff. So always interesting looking at immune genes and immune responses. This looking at this toll-like receptor gene, which obviously very, very important for immune responses. Did they post a phylogeny? I guess they didn't. Oh no. All right. Well, look, I guess for me, very, very interesting to look at this toll-like receptor, these toll-like receptor genes, and the specific different loci and the signals of selection, which are obviously looking at various things such as rates of, here we go, synonymous and non-synonymous substitutions, which would indicate that the site is under selection, or positive or negative selection, depending on which way you go. For me, I guess I would be interested to see other immune system genes, such as the major histocompatibility complex, which controls a lot of antigen binding and things like that, and a number of other immune genes. I guess this toll-like receptor must be super important for a lot of genes in birds, but that's an assumption. So I'm not entirely sure. Do you personally know what genes specifically seem to have been shown to be important for disease resistance in birds, or is that kind of genetic work yet to be done?
I'm not too familiar with the immune side of things because I just didn't have time during my PhD to look at it, apart from looking at some antibodies, as much as I would have loved to do more. But because of the large fieldwork component, there's always a limited scope.
Yeah,
but
there's never enough
time.
time.
You just showed, exactly. But the study you just showed is really interesting. And I think it really shows what I also mentioned before, that threatened species are particularly at risk, not just because we can't lose any of those remaining individuals, but also because they have this increased susceptibility because their immune genes are not as diverse anymore, and the immune system is just not as great as in a genetically diverse large population.
Yeah, it's kind of interesting because you think of like diseases coming through and you survive it, so you gain an antibody response, but on a population level, if things are being wiped out and only certain individuals which have maybe a very small subset of those immune gene mutations, then you're really kind of bottlenecking the diversity of those immune genes in that population and reducing the overall future ability of that population to adapt, possibly.
Absolutely right. And also, if you have a large population and a virus or other pathogen has been circulating in that population for a long time, there may be some coevolution, or the host may at least be used to that type of pathogen. There are antibodies there, maybe at low levels. They're just, to put it very simply, more used to that pathogen already. Whereas if you have a small remaining population, that may mean that it was not exposed to that pathogen for quite a long time. And if a pathogen then gets back into that population, it can have a much larger impact because these individuals are just not used to that pathogen anymore.
Yeah, absolutely. And with that wider distribution of a more common species, you can have pockets of locally adapted populations where different alleles can vary in their frequency, and you can have that extra genetic diversity, which small populations and particularly contracted and bottlenecked populations that have gone through selective sweeps are very unlikely to have.
That's exactly right. Yeah. And I mean, I think maybe that could be one potential question to further look at with Beak and Feather Disease virus as well, because that virus originates in Australasia. So our birds here maybe in general fairly used to it because it's been around for a very long time, but the virus is now spread around the world due to the pet trade mainly, so export of pet parrots. And if it then gets into populations that don't know that virus, it could have a more severe impact with more signs of disease, for example, as well. There are a lot of cool studies around worldwide, not too many in wild birds, so there's definitely a lot more that we could look at, and it's very interesting to see, but also worrying, and definitely good to keep an eye on it.
Right. Those studies from other parts of the world, are there many looking at genetics and genetic diversity, or what clades of beak and feather virus are more prevalent abroad, or not really?
There are a few studies, but I think the largest and most studies are really from Australia. There's a really cool paper by a previous PhD student in my group, Justin Eastwood, who again looked at crimson rosellas, but crimson rosellas are really cool to look at because it's actually a species complex. So I only looked at the really red crimson rosellas around Geelong, but he looked at all the different subspecies and also hybrids in this kind of ring species complex. And he could show that, if I remember correctly, he could show that the prevalence is lower in hybrids or in certain subspecies. And he linked that to evolution and speciation as well, and how genetic variety and the rarity of genotypes can contribute to differences in prevalence and impact of a pathogen, which is super cool as well.
Yeah, wow, fascinating. Well, look, we better move on for now. We've got some more new papers to get to. And let us go here once again. And another one of your own papers from your lab group, this one in Nature Scientific Reports. And we have it right here. Seasonal fluctuation of beak and feather disease virus infection in wild crimson rosellas. So interesting stuff. So we obviously know that you guys have been studying these birds for a while. And with that long-term study that you guys have going, you can obviously look at some fairly interesting things that you wouldn't be able to look at with a short-term study, such as fluctuations throughout not just one season, but I imagine multiple. So let us go quickly through the abstract here. Understanding patterns of pathogen emergence can help identify mechanisms involved in transmission dynamics. Beak and feather disease virus poses a major threat worldwide to wild and captive parrots. Yet data from wild birds on seasonal fluctuations in prevalence and infection intensity, and thereby the potential high-risk times for virus transmission, have been lacking. We screened wild crimson rosellas for BFDV in blood and cloacal swabs. Prevalence in blood samples and cloacal swabs, as well as viral load varied with Julian date and in blood, were highest after the breeding season. Breeding birds had lower viral load and lower BFDV prevalence in blood than non-breeding birds. Interesting. BFDV prevalence was much higher in younger, so that's less than 3-year-old, than older birds for both blood samples and cloacal swabs. Status in blood and cloacal samples was not correlated within individuals. BFDV status in blood and cloacal samples was not correlated in individuals. Very interesting. We show that at least in P. elegans, BFDV infection seems to occur year-round with seasonal changes in prevalence and load found in our samples. Our analysis suggests that seasonal changes were associated primarily with the breeding season. We also discuss age and sex as an important predictor of BFDV infection. So interesting to look at this seasonality as well. I guess I wanted to ask, what does it mean for you guys to see that there's no necessary correlation within individuals between blood and cloacal samples? So doesn't necessarily mean you're going to find it in both areas.
It can mean several things. So just to explain it a bit more in detail. So in some birds, we found BFDV only in blood samples, but not in cloacal swabs. In others, they were only BFDV positive in cloacal swabs, not in blood. And some others were positive in both sample types. And being BFDV positive in blood shows us that, yeah, the virus or viral DNA is present in blood, which we assume is mostly current infection. Whereas if we pick it up in cloacal swabs, it can mean several things. So first, we have to take that a bit with a pinch of salt, because we need to validate that method a bit more extensively. We also discussed that in that paper. I think it's a really interesting method to look at possible excretion of the virus, shedding into the environment via feces, but we have to have a closer look at whether what's actually excreted is viable virus or just these fragments I was talking about earlier. Another thing it could mean is that the virus replicates in various organs, for example, in the so-called bursa, which is an organ that sits very close to the cloaca and is important for immune function. So it could be that we pick up virus in cloacal swabs because it's hanging out in the bursa even when it's not too active and then not circulating in the blood. So it's a pretty complex pattern we see there, but I think it's quite intriguing because, yeah, some birds were only positive in cloacal swabs, but not in blood, and we wouldn't have picked them up as infected if we just checked blood samples.
Yeah, right. Nothing's ever easy, is it? can't
No.
just be one way. But
Especially
yeah,
not in the wild.
yeah, no, absolutely. And interesting as well that breeding birds seem to have a much higher prevalence.
Breeding birds were lower, yeah.
Lower, sorry, yeah.
Yeah, like I mentioned earlier, that came as a bit of a surprise to me, although we saw that in previous studies as well. My colleague that I mentioned before, Justin Eastwood, found something similar. But yeah, it's amazing because you would think that breeding birds may be stressed and they may be more susceptible to disease, and that's been shown for other parasites, for example. But yeah, I think what could be one of many factors is, again, that during the breeding season, the birds don't socialize as much. In winter, they hang out together in flocks, especially the young sub-adults that are also then showing the highest prevalence. And then during the breeding season, the birds split up into pairs and just hang around in these pairs and, with the finding food and incubating the eggs and so on and so forth. So that may reduce the risk of getting infected a bit. But that's speculation, so that's really hard to look at in the wild, really.
Yeah, definitely. So look, social distancing is good, but you are allowed to hang with your significant partner, and
exactly,
yeah, right. I guess another thing that I was thinking, and this is obviously very hard to test, but maybe the idea of upregulation of certain physiological systems during gestation or reproduction or nesting. I would not put it in the realms of impossible that things which are getting ready to reproduce or having young are going to maybe crank up the dial on certain immune responses or things like that, just to during that very, very vulnerable period for their young or their developing young, maybe buffer from some regular immune responses. I'm not sure if that's true at all, but it would almost seem to make sense as an adaptive response that, hey, during pregnancy or during nesting might be a good time to really, really crank up the immune response.
Would be really interesting to look at. Another option or another possibility would be that sick birds, weak birds don't even breed.
Yeah.
So only the really fit ones start nesting, and they may be less susceptible or just not infected. Could also be. So there are quite a few explanations. And it's just hard to disentangle. So we found that we had this really high, this peak time in our seasonal fluctuations after the breeding season, but that seemed to be largely connected to birds outside the breeding season having this really high prevalence, and also the young birds having this really high prevalence. So that, but in nestlings, we also had a really low prevalence. So chicks hatch and fledge, and then sometime afterwards they get infected and then suddenly have this massive prevalence, and then some probably die, some manage to clear the virus, like we showed in that paper you first mentioned, which then leads to this lower prevalence, and then the older birds. The great thing about Rosellas is that you can age them so easily because the young birds just have different color. So that really helps with that study.
Yeah, nice. So I guess just in terms of management implications for the disease, I guess we know when the bigger risk time for a bloom in this disease in Rosellas is, and what might that mean for co-occurring threatened species?
I think hopefully it can help with a more targeted management, just to focus at that time of the year in protecting endangered species, I guess particularly well, although they already should be protected very well if possible. And also to keep an eye on especially young birds, because that young birds are particularly susceptible to BFDV and have this high prevalence, has been shown in other species all over the world as well. So I think that could maybe be one way forward to really target the protection of the young individuals of the population as well.
Interesting stuff. Very cool. Well, look, I think we pretty much have to wrap it up there, unfortunately. Man, is there anything regarding beak and feather disease that we haven't covered that you think is important for people to know at this stage?
I don't really think so. Apart from really keeping an eye out and, you know, cleaning well, and also helping to protect the environment and protect the birds, to take a bit of stress away from them so that the disease doesn't spread as readily.
Yeah, for sure. And I guess in terms of reporting the disease to local vets, local wildlife carers, is there any groups that you recommend?
There's not that much really around. I think one thing that's possible is if a sick bird comes in to your feeder, maybe stop feeding for a while to just give the birds a bit more of a chance at social distancing, so that that sick bird doesn't spread the disease around. If the bird is in super bad condition, you can try and catch it or ring a wildlife carer to catch it and bring it to a vet. But yeah, there's not too much we can do. There's no treatment.
Yeah, right. So keeping an eye on your birds and monitoring and keeping things clean, always the best way to go. And social distancing, we're all learning more and more about it these days.
Yes, and environmental contamination, keeping surfaces clean, cleaning your hands after you touch things.
Yeah.
Yep.
Absolutely. Well, Johanna, thank you so much for joining us. This has been super fascinating. If you guys come out with some more research in the future, we'd definitely love to talk to you again if we get the chance. And mostly stay safe down there. We know that it's a little bit tough down in Victoria in your area at the moment. So yeah, glad to see that you're healthy and well and staying safe. Thanks so much. Thanks for having me. It was good fun to chat. No worries. Thanks for joining us. Everybody, that's Johanne Martens. You can find her on Twitter at Johanna_Martens, or you can follow the research at Deakin Center for Integrative Ecology at Deakin CIE. We'll be back with plenty more wildlife cake and cocktails shortly. See you soon. See you.